Okay, in this video I'm going to go over types of transport. I feel like some people have like a lot of difficulty with this and don't get it that much, so I'll do my best to explain it. And then, honestly, this is pretty much common sense, but a cell must exchange materials with its surroundings, and that's controlled by the plasma membrane because it surrounds the cell and controls what goes in and out. And hydrophobic molecules, since the middle of the cell membrane is hydrophobic, it's pretty easy to pass through. But polar molecules that like water do not cross the membrane easily because they have to cross through like a whole forest of hydrophobic stuff and they just don't get along. And then I was talking about the big proteins in the membrane in the last video, and most of them are transport proteins. They allow passage of hydrophilic substances across the membrane because, like I said earlier, hydrophilic substances can't go just through the membrane because either they're too big or the charge, it just doesn't work out. And some transport proteins have channel protein means it's just a straight line and they could just go there like a tunnel. And a special type for the passage of water is an aquaporin. Other transport proteins called carrier proteins change shape to let the molecule pass through, and I'll show you in just a second what that looks like. So this is a channel protein. You see the solute goes in, it's just like a tunnel in like a road. You don't have, it doesn't have to change shape because it just goes in and out. But a carrier protein, it goes in and it changes shape to go the other way, and then you can transfer in and out of the cell. I mean, this is just an example of one. Each of them look differently for different solutes, so it's just a wide variety. And we talked about diffusion earlier. I think I talked about it in one of my other videos. It just happens from high to low, but some molecules, like I said, are too big or are polar, so they can't go diffuse through the cell membrane by themselves, so they need the help of something else, like a protein, and that's called facilitated diffusion because the protein facilitates the diffusion. For example, I talked about it earlier, aquaporins, the channel proteins for water, are a facilitated diffusion of water. And ion channels that open or close in response to stimulus are also um, facilitated diffusion, and this doesn't use energy because it's still going from high to low concentration. And then, like I said, it's still passive because it doesn't use any energy. Um, I'm pretty sure there's an example of this at the end or a picture of it, and I'll show you that later. But active transport, it moves substances against the concentration gradient, so from low to high, and that you need energy to do that because it's not natural. And the energy comes in the form of ATP, and it's performed by specific proteins, and I'll show you them right now. And the most common one for us, type of active transport, is the sodium potassium pump. So basically, we want less sodium in the cell and more potassium, but that's going against its concentration gradient. So the sodium molecules go in these little circles right here because they're circles. ATP comes in and changes the shape so that the holes are facing the outside. The Na escapes from here, K plus goes in, and then it changes back. The shape changes back to this shape, and the K plus goes, and it starts over and over again. So every time it opens and closes, it needs ATP. Again, the sodium potassium pump, as I showed before, is one. And here are the steps. See, the sodium goes in, ATP changes to ADP, and the extra phosphorus goes and gives the channel energy. It changes shape to face the outside. The Na goes out, and the Ks come in. Again, energy is then lost, and it shapes back on the inside. K's go away, and the process starts over again. It's just a big circle. And basically, one of the most important things is that ATP is a source of energy. And again, diffusion, it just goes in and out, and the molecules have to be non-polar and small for just some diffusion to happen because it has to pass through the membrane by itself. But polar or big molecules, they can't pass through with regular diffusion, so they need facilitated diffusion. And like I said, there's the channel proteins and the carrier proteins, and these two different types help these molecules pass through. But active transport, it needs ATP because you're pushing it against its concentration gradient from low concentration to high concentration. And then she said this is really important, so pay attention. Membrane potential is a voltage difference across a membrane. Like, if there's more positive stuff on one side and less positive stuff on the other side, the one with the more positive stuff is going to be positive, the one with the less positive stuff is going to be negative, and this voltage creates something that the solutes can go down, and that's called the electrochemical gradient, because the chemical force is the ion's concentration gradient, so going from high to low and low to high. And there's also an electrical force because positive molecules want to go to the negative side of the membrane, and negative molecules want to go to the positive side of the membrane. So if you try to go against that, that's also going against the gradient, so it also is a form of active transport. And again, one of the basic types of electrogenic pumps that use a voltage is the proton pump, and I'll just show it to you here. So this is active transport because there's ATP. H plus goes out, and it needs ATP because it's low to high concentration. And since there's more H plus on the outside's going to be positive, and the inside's going to be negative. So not only is H plus going against its concentration gradient from low to high, it's going against its chemical gradient because H plus wants to go to the negative, but it's stuck next to the positive. And a special type of transport is cotransport, which means that it has active and passive transport in it. And here's the best way to show you as an example. So like I said, we showed you the hydrogen ion pump in the last couple slides. So the hydrogen ion goes out, and that uses energy, right? But the hydrogen wants to go back into the cell because I told you the concentration gradient and the chemical gradient favors it going inside. So it diffuses inside, and the sucrose just tags along with the H plus, and sucrose gets to go inside too. So it has active transport to get the H plus out and sucrose to get it in. And it's called cotransport because one is going um, towards its electrochemical gradient and one is going against. So co means two, or working together, and the H plus and sucrose are working together for the cell's well-being. And the last type of transport is bulk transport, and there's two types: exocytosis and endocytosis. Really large molecules, they just can't fit through the cell membrane no matter what protein and whatever, so they need bulk transport, and bulk transport requires energy because it's like a lot of stuff. And since there's no great picture of this, I'm just going to explain it to you using this picture. So basically, this plasma membrane forms a little bubble around whatever you're transporting, and the bubble goes in or out. And so it's not passing through the plasma membrane, it's passing in the plasma membrane. And in exocytosis, the vesicles, which are the bubbles, go take molecules from inside the cell outside. Exo, exit. Quick way to remember it. So as you can see, there's a vesicle, a plasma membrane around some molecules that need to go out. So it's just going to go travel along its way, break through the plasma membrane, and the plasma membrane is going to let out these molecules, and the bubble around the molecules is going to become part of the outer plasma membrane. And endocytosis is the opposite of exocytosis. It's taking molecules into the cell. And there's three major types. There's phagocytosis, which is eating, which takes in nutrients. There's pinocytosis, which is drinking, which takes in fluids and all the solutes dissolved in those fluids. And there's receptor-mediated endocytosis, and that's just for like chemical signals and stuff. And in phagocytosis, a cell engulfs a particle because it's nutrients. And so the plasma membrane makes a circle around the molecule, brings it in, the plasma membrane fills itself, and it does its stuff inside the cell. In pinocytosis, the same thing happens, but it's just instead of a nutrient in the vesicle, there's some water. and some other fluids. And in receptor mediated, um, the binding of ligands to receptors, so basically like a Lego to another Lego, if you want to think about it like pieces in a puzzle, makes the vesicle happen. And here's just a really good um, picture to explain that. As you can see, phagocytosis, food is coming in, the plasma membrane's going around, and here's the vesicle once it's finished. And the same thing with pinocytosis, except it's fluids. And receptor-mediated endocytosis, here are the receptors and the triangles are the ligands. So when the triangles go into the Ys, it connects the piece, and that makes the plasma membrane want to separate. And that's pretty much it for transport. So good luck on your quiz on Monday, and make sure you study the video and the types of cell structures too.